in the river, actually at the Bakersfield Environmental Studies area, right across from the university. And in the video, you saw hundreds of trapped fish. And what we observed and were not prepared for were thousands of fish. In some parts, you would see just massive piles like this. Water just disappeared. Some of them are still twitching or glistening. And we documented over 3,000 dead fish in 1.5 miles, which is about how long the river is at the Bakersfield Environmental Studies area. And so this happened because of a rapid dewatering event that we actually didn't know was going to happen when we started studying the Kern River more thoroughly. And so just for kind of some visual examples here, especially for those who are not locals. So August 23rd, this is a Friday. This is the Kern River. We're just right by the Stockdale Bridge. And this is the 1 week later on August 30th. We showed up and pretty much all the water was gone from our most downstream site. If you're familiar, this is right next to Riverwalk, and so here's the pedestrian bridge above the Bellevue Weir. And this is just a week of time before and after the dewatering event. And so this is really what I'm going to spend a lot of time talking about, is how dewatering of the Kern River affects the river ecosystem. Before we dive into more information and some findings related to that, just some little history or background information on the Kern River. It begins in the Sierras, and much of it is a perennial river. And a lot of the water comes from stormwater, snowmelt, and there's also Isabella Lake where the dam is. And so the Lower Kern River is from the lake that goes all the way through the canyon and in through Bakersfield. That's the Lower Kern River, and that's really the part that we focus on in terms of our research. And so parts of the river are actually designated as wild and scenic, and also runs through several parks and wilderness areas, and is very popular for recreation, such as fishing and kayaking. There's diverse wildlife that lives in the river and also around the river. And some of these are endemic or protected and endangered species that are found here or just in California, or specifically in the Kern River. For example, like the little Kern golden trout. And so there are a suite of anthropogenic activities, so human-based activities associated with the river. So for example, we divert water from the river to support needed agriculture for municipality use, okay? And again, a lot of the flow in the lower part of the Kern River is controlled by dam releases. And so my journey with the Kern River, the Lower Kern River in town, really began after I came here for my faculty position. And so what you're looking at are some of the very first students that joined my research lab when I first came here. This is Amy Fetters. She was a master's student. She's since graduated. And this is Caitlin Macardanis. She's a graduate student in the state of Washington. And we are hiking. So this is the Mohawk Bridge for those of you who are locals. And do not come from California. I'm from the Northeast and also the Great Lake area, very water-rich area. So I had never seen a river just like dry up so quickly before. It's extremely dynamic. And I never hiked a dry riverbed ever in my whole life until I came here. And so we actually were hiking the river to develop a research project for my previous grad student, Amy Fetters. And we started collecting data, and then a month later we showed up at our site and there was water there because there was a dam release from Isabella. And so this just kind of demonstrated how very dynamic water availability can be in the Kern River. Now, this was before the pandemic, and then what happened was during the pandemic, there was a lot of momentum that built up, especially with local activist groups and citizens that really wanted water to be in the river. And the reason is we lack a lot of water in the river because of water extraction. So sometimes the river is intentionally dewatered because of maybe repairs on weirs have to be done, or we're diverting massive amounts of water that turn it into basically this dry riverbed. And so during the pandemic, actually, this is an artist rendition here, and this is the Mohawk Bridge, just to kind of demonstrate the lack of flow that is expected to be in the Kern River that actually isn't there. And this, I believe, if I remember correctly, was commissioned by the group Bring Back the Kern, which is under the umbrella of the Kern River Parkway Foundation. This also brought awareness to the Lower Kern River. And so there's an organization in the United States called American Rivers, and every year they give a report on what they identify to be the 10 most degraded rivers in the United States. And in 2022, the Lower Kern River, the river that goes through our town, was ranked as the 7th most degraded river in all of the United States, okay, because of excessive water withdrawals. And so here you can see the dry Kern River bed and a biker on the Kern River bike path that goes alongside it. And so to continue to bring more awareness, the very first annual Kern River Festival happened in, I think if I remember correctly, that was May of 2024. And so here we developed a trash and Kern River education booth here. And I was visited by this gentleman here right at the beginning of the festival. And this is attorney Bill McKinnon from Water Audit California. And this gentleman literally got down on his knee and begged me to start studying the Kern River and do it like immediately. I had plans to develop more research in last fall because that was going to be a sabbatical. But this, I think, highlights the importance of trying to listen to the citizens and other concerned parties when there is, maybe an environmental, ecological degradation of concern. And so I worked with folks on his team to identify study sites that we started studying on a weekly basis during summer of 2024. And so this is a satellite image of Bakersfield and the surrounding area. And these blue represent some of the weirs that control the flow and water availability through town. And so we have our upstream site, which is Hart Park. This is above the weirs and right around, right just upstream of where first point of measurement is, if you're familiar with river measurement, okay? And essentially our control or reference site, if you will, because it doesn't really expose to much of the water diversions at this point before we hit a bunch of weirs in town. We then identified 4 downstream sites of 4 weirs that we have been sampling. We sampled last summer, so summer 2024, on a weekly basis, and since then on a monthly basis. So we actually have about 18 months of Lower Kern River data. And so just to kind of remind you, this is one of the photos I showed you in the beginning. This is our most downstream site, okay, the Bellevue Weir right next to the Stockdale Bridge, okay, when water was still flowing there in summer of 2024, okay. So, so this is before and after the dewatering event. So we were conducting weekly monitoring on the Kern River through July and August, and we did not know that there was a plan to actively dewater the river. So when we showed up, okay, at the end of August for our weekly monitoring, we were very shocked. And this created an interesting opportunity to study how rapid water loss and maybe long-term reduction of water availability affects a river ecosystem. Okay, so specifically, one of our research questions, how does water availability affect the Lower Kern River ecosystem? For the confines of this presentation, and along with the data that we are presenting, we predict that with a reduction of water availability, there would be less river habitats, and with less water, the water would actually get warmer. Okay, and warmer water typically holds less oxygen, so we expect there to be less oxygen available in the water to support aquatic wildlife. These collective changes in environmental conditions were expected to result in a drastic reduction in aquatic wildlife with, for example, aquatic macroinvertebrates like this dragonfly larva pictured here and fish community. And then we also expected that this would result in a shift in human direct interactions with the river. Okay, for example, like an increase in access directly to the river channel, which would increase potentially trash or anthropogenic litter in the river. And so, just briefly, some of our methods here on our weekly monitoring for summer of 2024 and monthly since the dewatering event. We have this sensor that has like pH sensor, oxygen, etc. So we measure a whole bunch of parameters of the water. We also measure depth at all of our sites. And then we take water samples sometimes to measure nutrients and other human chemical contaminants, and then wildlife observations such as the bird community, fish, and also the aquatic macroinvertebrates. For our fish, we do visual surveys. I'm pointing this out because I'm going to talk a bit about those data today. And we also do eDNA sampling. So E is for environmental DNA. Essentially, we take a water sample, concentrate the DNA, it gets sequenced, and then we can identify what fish are in the water based off of the DNA that's residing in the water and compare that to our visual observations. So basically, the rest of my presentation is broken up into this river sampling timeline associated with water availability. So we started monthly monitoring, okay, in July and August of 2024 when there was still a lot of water in the Kern River. And then by September 6th, rapid dewatering of the river had already started through multiple miles of the river channel. And then by the end of September, the river had been dewatered. And so I'm going to share with you some data and observations associated with these different like transitions of water availability in the river. But please note this data collection is still underway. We're still processing the data, and we have 17 months of data, so there's still so much to look at. So I'm just going to share a little bit of data with you, along with a lot of visual documentation for the story of the Kern River as it's been unfolding for us over the past year. So our first data slide here, we have our weekly monitoring, and so we've got week date here on the x-axis and the mean depth in centimeters of the water on the y-axis. And these lines are color-coded based off of our sites. So upstream is our control, Hart Park, above those weirs where water diversions are in town. And then as we go from cooler to warmer colors, we're coming downstream where more water diversions have been happening. So this red line is our most downstream site, downstream of the Bellevue Weir, just right next to the Stockdale Bridge. So overall, you can see that there's a significant reduction in water availability as we go from upstream to downstream. And we see a lack of data points on this part here for these two sites because that's when the dewatering of the river happened and there was no more flowing water for us to measure the depth. And so our Stockdale, our most downstream site, okay, this August 23rd is the last day we have water measurement. Okay, so again, this is what it looked like. And then a week later, we showed up and it was essentially completely dewatered with just one pool of water left. Now, if we focus on water temperature, and so the colors for the sites are the same, okay? And so water temperature is significantly different across sites and through time. And just for reference, okay, about 90 degrees Celsius is low risk for a lot of fish species, especially fish species that need more cooler waters. But 24 degrees Celsius or higher prolonged exposure increases health risk and stress of fish. And these values come from the Environmental Protection Agency. Okay, and so you can see that we definitely have temperatures that exceed this threshold, which means that maybe some of our cooler water fish won't be able to survive long term in the Lower Kern River unless there's really deep, cooler shaded habitats, okay, for these potential cooler water fish that could colonize the Lower Kern River. If we focus on this data point here, our warmest in this weekly monitoring period, we actually put out these temperature loggers that you can anchor in the river, and then it can record the temperature, for example, every 30 minutes. So you have like these day and night diel patterns of temperature. Okay, and we've got time here on the x-axis. So this is the temperature at our most downstream site over the span of a week. And that 24 upper threshold, all of these temperatures are above that threshold, which is very concerning, especially for fish that need cooler waters to survive. In terms of oxygen, what we observed was again a unique oxygen pattern with some of the oxygen oxygen availability decreasing as we go from upstream to downstream. And something I just want to put here, this is percent oxygen in the water, is actually while we see this decrease in oxygen across the board, on average, overall, many of the measurements are above 100%, which means that sometimes the Kern River is hypersaturated with oxygen, okay? Which is important because wildlife need oxygen, right, to breathe, to survive. Why we think this is happening, at least during the day, is because there is a lot, well, there was a lot of aquatic vegetation. So you're looking at floating aquatic vegetation. A lot of it is invasive species. So like with the purple flowers, that's water hyacinth. With the yellow flowers, that's floating primrose willow. There's also a lot of rooted aquatic vegetation. It's like an underwater forest in some parts of the river. And plants add more oxygen to a system. And so actually I just presented some of these data at the California EPA headquarters, and someone recommended we take oxygen measurements at night because it might be that at night there might be a lot less oxygen since plants are not very aren't producing oxygen really at night. And so that's something we're going to explore in the future. And so if we were to average those oxygen values across the summertime, we do see a significant reduction in oxygen availability in the river water as we go from upstream control to our downstream sites. And so again, with our weekly monitoring, so that's some of our data there, but we also have a lot of observations. So we saw the typical habitats that you would expect in a river with these riffle and run and pool habitats, some pools in the river. So I'm 5'6". This is actually, it's hard to tell as a pool. This is a willow tree, but it's actually about 6 to 8 feet deep here. It's a snorkeling pool. And there's other pools that are at least 10 to 15 feet deep in the Kern River as well. We also observed a plethora of aquatic macroinvertebrates, which was really exciting to see. I'm actually an aquatic entomologist by training. And so here you see this critter crawling. That's a damselfly larva. Okay, we've got caddisflies. We also have clams. We also have mayflies and snails. And macroinvertebrates are important to study because they are an honest signal of how healthy a system is. We are still waiting for our scientific collector's permit from the state of California to do some formal monitoring of these critters. These are field observations, but finding caddisflies and mayflies is really nice because they tend to be more sensitive to pollution. So having them in the river right now suggests that maybe some of the water quality is good for some of these more sensitive macroinvertebrate species. We of course observed, these are underwater photos, that's why they look a bit darker. Lots of fish as well. And so for example, we've got juvenile and adult bass and sunfish, bluegill sunfish here. And so I'm going to talk more about fish in just a little bit there. And so, as I mentioned, we were doing our regular weekly monitoring, and we showed up actually with a reporter. If anyone knows Lois Henry with SJV Water, she asked to come in the field with us, and she picked the best day because we showed up and didn't even know the river started being dewatered, and she got our raw reactions. Okay, so I'm going to talk about the transitioning of the dewater, which started happening just very briefly over a couple days to a week. And so before the rapid dewatering of the river, we had been noticing that the water level in the river was being reduced throughout the summertime. This created these refugia pools that trapped aquatic wildlife like tadpoles and fish. That you can see that these pools are evaporating over time, and fry fish, juvenile fish, and other fish would get trapped. Okay, and we have these algal blooms that were happening as well. So this is before rapid dewatering of the river. This is when the river water was kind of like slowly decreasing throughout the summertime, which also meant some sandbars and alluvial islands were also being exposed as well through time. And so when the river was being rapidly dewatered, it went from being a flowing river to more disconnected habitats. So the water became more stagnant and the habitats became more marsh-like. And you had a lot of these refugia pools that had high concentrations of aquatic wildlife because there was no connecting water from pool to pool. So you had like hundreds of fish, for example, in one pool, and they were just trapped there. And so this is example of what I'm talking about. This is when we showed, this is the Stockdale Bridge, okay? And we showed up with the reporter, and this is my currently he's a graduate student, but at the time he was an undergrad, Andrew Alba. He's one of my lead students on this project. Is we showed up and we were just shocked because it was like someone snapped their fingers and all this water was gone. And so we have a refugia pool here, okay? I have a video here, so this is from the other side. But you can see there's actually hundreds of stranded and dead fish. You can see the big ones like carp and channel catfish and bass, okay? But there's also, it's a little bit harder to see, really small juvenile fish as well. You can see some are actually still twitching as well, okay? And we were unprepared. We were unprepared for this, okay? Some are still trying to breathe. And in that pool, some of them had actually started to get picked at by birds, and there were lots of dead carp and other fish. But there were about, I think, if I remember correctly, about 60 live fish, and these were only carp were still alive. This pool was about 15 centimeters of water, had extremely low oxygen, and the water temperature was very warm. So, and they had already started to shed their scales, which suggests the fish were under extreme stress. So we had actually planned to do a visual transect fish survey at the Bakersfield Environmental Studies area. And so here's Stockdale Highway, and CSUB is just right here, just below the highway, just south of it. And north, so this whole floodplain area, this is the BISA. From Coffee Road, there's the weir right here, the River Canal Weir, all the way down to Callaway. It's about 1.5 river miles, okay, if you follow the curvature of the river. And so essentially what we did is we went in to the river, and so it was me, a team of current students and alumni, and also the reporter, Lois Henrich, was there, and Dr. Antia Lauer, she also had joined us as well. We got into a line and we just like marched forward and wrote down observations of fish and other wildlife that we observed. And so these are just some more photos. So the reporter, she had a drone, so here's like a drone footage of our aerial photo. And so we've found lots of fish, a couple of dead fish, but nothing more than you would typically expect. And this is a video, so this is a school of juvenile bass, just like what's pictured here. And there's a little divot in the sand, so their schooling together helps reduce drag. I point this out because I have, want you to remember this, okay? Because I'm going to come back to it. And so also during the summertime, before dewatering happened, okay, we took those water samples for fish environmental DNA. And what we have here is our field observation. So before the river, the Bakersfield Environmental Studies area was dewatered, which is upstream, our next site upstream of the Stockdale Bridge, where dewatering first happened. We observed lots of fish, okay, and this, our most common were like carp, okay, mosquito fish, bass, and sunfish. And it turns out that the environmental DNA and observations throughout our sites from the summertime aligned nearly beautifully, which was very exciting for someone who's not like a fish biologist, okay? And so some taxa of interest, one is the Sacramento sucker, which is a native fish. Most of the fish we see in the Kern River are not native. The eDNA also picked up threadfin shad, which is one we did not observe in the field, or if we did, we missed it. Another that was really interesting is rainbow trout. We have observed rainbow trout in the Lower Kern River multiple times, from Hart Park all the way down to the Bakersfield Environmental Study, not Hart Park, excuse me, Panorama Vista Preserve, okay, which is our first site downstream of the weirs, all the way down to the Bakersfield Environmental Studies area. And we are not the only ones. Other faculty, as well as local citizens, have said to us that they have observed the rainbow trout. And so the trout that we have observed, we have only observed juveniles. Okay, it could be that some of these trout are maybe hybrids, maybe left over. Some of any adults people might see might be left over from fish plantings, but me personally, I've only observed juvenile trout. Okay. It was not picked up in the eDNA, and this actually is not surprising to us. It's because you oftentimes need a lot of DNA in the environment to be detected, and you need to be there at the right time at the right place, or you can miss it. So it could be that the rainbow trout population in the Lower Kern River is really low, and so maybe there's less DNA available in the environment for it to be detected. Okay, so we talked about our weekly monitoring before dewatering, transitioning of dewatering, and what happened. Now let's kind of transition into what we've been doing since the river was actively dewatered. And so what we saw is probably what you expected, is the water was all gone eventually. And so there was no riffle run or pool habitats, no marshy, there's essentially no water in major regions of the river. And so I think if I remember correctly, about at least 6 miles of the river had been dewatered. Okay, and we saw lots of increase of anthropogenic litter and human trash, lots more of dead fish, and just dry exposed riverbed. And so this is the river canal weir right there at Coffee Road. It's actually, if you are local and you drive by, it's been replaced. They pulled it out and they put in a new one that they just started turning it on just the other week. And so that controls flow into the Bakersfield Environmental Studies area, part of the river. And so when the rapid dewatering extended up to the BESA, the Bakersfield Environmental Studies area, we rushed to go ahead and do another fish transect survey. And so this was a week apart. So we did our quote live water fish transect survey. And then a week later, we went back out and we rehiked the whole area again. And in my lab, the students call it the dead fish transect survey because all the fish were dead. Okay. And so we would hike, and then if we saw fish, we would put our meter stick down to take photos. We took hundreds and hundreds and hundreds and hundreds of photos that are still being processed. So we have a ruler, we have a computer program, we can measure the length and use this information for identification and also to classify into age class, and how the adults, juveniles, etc. Okay, and so for example, we saw piles and piles of dead fish, kind of similar to what we saw at the Stockdale Bridge. And so here's our BESA site again, just so you can see. So we wiped the whole area. All right, so here's the dry river bed and team of folks helping with this. Okay, and this is what it looked like. So here's the dry river bed, and this is a week before when there was water there. Okay, so drastic change in just 7 days. Okay, and so here's just more of piles of fish, okay, for photographs that are being processed for data collection. And so I asked you to remember the school of juvenile bass here in this little divot. So here's this again, right here. And it's a little hard to see in the video. I don't think is working right now, but these little brown bumps, those are like the dead fish. They're trapped there because water, it's a little bit deeper. There's probably a little bit of residual water, and they were trapped there, and eventually they all died because water didn't come back fast enough. Okay, so here's, for example, a bluegill sunfish, all right, that we would measure with a computer. And sometimes they were separated like this, or they were highly concentrated like this, where you could have 100 fish in a photo. And this is the video I showed you before. So we've got fish that are trapped on their side in this very shallow pool, hundreds and hundreds of mosquito fish, as well as tadpoles and juvenile bullheads as well. And so I want to return back to our fish data, but specifically for the Bakersfield Environmental Studies area. The table I showed you before was really linked for the whole river system, part of the system that we've been studying. But if we focus on just the BISA, these X's are the fish that we observed when we did our live or water flowing transect survey, and then the dead fish essentially. And so unsurprisingly, we found more species and more numbers of fish, because when you're a dead fish, it's not like you can swim away from us, right? You kind of like just stuck there. All right, and our eDNA, specifically samples at the BISA, did add a couple of taxa that we missed. So collectively, all three of these help to boost overall our fish data, these combined strategies, which is really nice. We've also expanded this to the Panorama Vista Preserve. And so actually since September through most of October, we've done weekly fish surveys, because the Panorama Vista Preserve, our one of our more upstream sites, started to be dewatered, but it never got fully dewatered, and so we just have flowing in eDNA. But it aligns very nicely with what we saw downstream at the BISA. And so some just preliminary data of the fish community. This is based off of our dewatered transect survey, so when the fish were dead. Okay. Overall, we saw a lot of western mosquito fish and a lot of sunfish and bass. A lot of those organisms, okay, were zooplanktivores, so they eat a lot of plankton in the water. Zoobenthivores, so they eat a lot of those aquatic macroinvertebrates I showed you photos of before. And most of them, in some cases 100% for some of the taxa, were juvenile fish, okay, with larva fish being the rarest life stage. And so these data collectively have been presented multiple times. And so these are some students who presented on some of our Kern River fish data and algal data at the CSUB BC Summer Research Program. And then the fish data set is really led by my undergrad student, Stephanie Flores. And so she presented this poster just the other week at the California EPA headquarters. There's the California Water Board Biomonitoring conference that happens every year. So she presented there. And I do want to say that she's the one processing those hundreds of photos of thousands of fish. She's measured over 2,000 fish to generate the data here. And she has about 1,000 more to go. She's very excited about it. I'm being facetious, because we find more dead fish just this past week. Every time we find, she's got a lot more fish measurements to go. All right, so as I mentioned, when we went out, the reporter, Lois Henry, she was with us, and so she was the first one to report on our research activities for the Lower Kern River, and this also generated over 30 news articles, and we made the front page of the LA Times as well. And then I also went to the City Public Hall meeting last year in September, and this is a public letter that I read to the city council, and I gave them all a copy to let them know we preliminarily counted over 3,000 dead fish and just that had died in just a week at the Bakersfield Environmental Studies area. Concerns due to the rapid dewatering of the river. And so few other observations and information to share with you that are developing and continuing projects for the Kern River. We've been studying trash in the river on and off for quite a while now, but something I wasn't thinking about at the time, which is really funny because I studied plastic pollution in the environment, was I was not prepared for the amount of trash that just like showed up so quickly after the river had been dewatered. And so a substantial increase in a lot of trash in the floodplain area and in the river channel itself. We also saw pop-ups of lots more tents. And upon reflection, this actually makes a lot of sense, because we do have an unhoused and transient community that live along the river and literally in the river, like on some of those alluvial islands. And unlike the fish are trapped. The fish can't swim upstream when the water is receding, because the weirs are a physical barrier. We do not have any fish passage. So once a fish goes downstream, they're trapped in that part of the Kern River unless they keep going downstream. But humans, we can migrate. And so as the water level was, as water was receding from downstream to upstream, humans were relocating where they were living and abandoning some of their items they had collected, and then collected new items, and then would abandon or take some of them with it and then keep going. And so we saw this all throughout last year and fall semester, and also more recently as water has started to return to the Kern River. And so just a little sneak peek preview, okay? We actually have 4 bridge sites from Mohawk Bridge, Callaway Bridge, Stockdale Bridge, and Allen. And so CSUB is right here, our university. Here's the Bakersfield Environmental Studies area that we've been monitoring trash at. And so we actually collect trash across the river and transects above and below bridges, under bridges, at these piers where debris dams form. We bring it all back to the lab, clean it, wash it, and do a bunch of measurements. Okay. And so this research has been developing even more recently because of the change in water availability. And so this past summer, I was part of the CSUB or BC 2025 Summer Research Program. So these are the students that were part of this in collaboration with BC Professor Milena. And we went out and surveyed all of these bridges for anthropogenic litter and brought it back. And so just one slide of preliminary results here. You see this bright pink? Okay, that's plastic. Plastic is the most dominant form of material that we find in the river. And some of our sites and habitats have more variety of anthropogenic litter materials than other sites. Okay. Now, there's a lot more data associated with this, and some of our students from the BC CSUB Summer Research Program presented it, but this is actually being led by my undergrad student, Dana Garcia. She's gotten funding from a STEMnet and CSUB SRS program, and she actually just collaborated and led with me another sampling event just this past weekend, this past Friday, Saturday, Sunday. And again, a lot of BC students came and helped, as well as CSUB students. We went back to these bridges and we collected our trash, which is now at our ESA site on campus, waiting for us to process it. Thank you, Melina, for thank you. I appreciate that. All right, and so this is important because water started to return to the Kern River. And for the first time in about 13 months, there was water flowing from Hart Park all the way down to Allen Bridge, okay, just a couple weeks ago. And so as the river canal weir at the Bakersfield Environmental Studies area at Coffee Road, that had a ribbon cutting ceremony and they let water flow. And then we had rain events a couple weeks ago. They had to do dam releases. And it's the first time that we had a fully flowing river of some meaningful flow in 13 months. And what you're looking at here is all this trash. And so as humans have gone into the river bed over the past 13 months, where some parts have had water, some parts have not. This is the Bakersfield Environmental Studies area, and it's been dry for 13 months. And so while we saw water is here, we were out a few days later. How quickly does wildlife recolonize when water comes back, like macroinvertebrates and fish? And while hiking in our BISA, we saw all this trash. And so these are some of the students, but you can see all this trash around them. And water flow plays a really important aspect of the distribution of human trash in river systems. And so we just collected these data after the water level has gone down in the river. So there's been water in the river for 2 weeks. And so now we can ask a new question. Well, how does the water availability affect anthropogenic litter distribution in the river, which is what Dana is going to continue to explore. And so just to summarize here, the big picture of what I've shared with you today is when water is present in the Kern River, it's pretty logical, right? We have a diversity of riffle, run, and pool habitats that support a wide variety of aquatic wildlife, as well as terrestrial and semi-aquatic wildlife. So we see more, more canid animals present and mesocarnivores because water is present. We also see a lot more birds like herons and egrets and ducks when water is present. And with the reduction of water availability, this really shifts the aquatic habitats, which in turn, of course, are going to have a massive impact on wildlife that live in the river system. And if all the water goes, we end up with that dry riverbed, which we often see here in town throughout the year. And so, excitingly, when water came back just 2 weeks ago, my team, as I said, we went out and we didn't see any fish. Doesn't mean that they weren't there, but we found giant crayfish. We saw the return of riffle, run, and pool habitats. And my video's not paying, but we actually saw a lot of dragonfly adults laying eggs just a few days after water returned to the Bakersfield Environmental Studies area. And so that was pretty exciting. And so next steps, we are continuing to study the river and develop projects and work with community engagement and science communication. So for example, some of us have participated in the Kern River March, and there was a second river festival this past May. There's also a lawsuit happening with the Kern River. And so I've been making some of the data available so any concerned citizen or any person could download it from the website. We are behind though. I still have a lot more data to upload to the website. But that's important because there's U.S. Fish and Game Code 5937. I'm not an expert, but my understanding is that owners and operators of dams need to maintain water downstream for fish to be in good condition. And so if you've been following in the news, there's a lawsuit associated with the Kern River and also Kerns that California public trust doctrine has been violated. And so we've been in conversations with attorneys, answering their questions and also considering the type of information that they might be available, might be useful for them. Okay. And then, of course, river restoration. And so there are other rivers in California that have also gone through a similar type of lawsuit and also needed legal regulation of water to be required. And there's another organization, River Partners, who's been developing a riparian Kern River restoration plan, which I think are going to start executing next year in the spring, which is pretty exciting. I've got this slide with here. This is just some information about that lawsuit, some of the major steps associated with that. But parts of the lawsuit, actually, I think if I remember correctly, are going to be heard by the California State Supreme Court. But I think our local judge court hearing is delayed until I think like 2027. I may be wrong about that, but you can look it up in the news. Okay, so I would like to acknowledge, that the land that we conduct our research with is home to many indigenous communities, and we promise to continue to be conscientious stewards of the land for future generations, and hope that our research activities support the goals and missions of these communities as well. Also, these photos don't do any justice for the number of students. I think over 50 students have helped out on this research over the past year. And I really appreciate my students' help coming out with me a lot of Saturdays and Fridays to survey the Kern River. And so acknowledgement for lots of help and support, as well as funding from various CSU and CSUB sources. And we really appreciate all of that help and assistance. And so with that, I'm happy to take any questions. Thank you.
Thank you so much, Rae. This is a fantastic talk, and it's amazing research that studied the ecosystem along the Kern River. So I learned a lot. So that's really fantastic. There's a lot of questions actually on the chat, as you can see over there. I moved some of the questions on the Padlet, but David asked a couple of questions. One is about the pond at Riverwalk. So he's wondering if the pond at the Riverwalk are stocked with fish, and if so, what species are put in the pond on the Riverwalk? Will this fish swim back and forth between the river?
Yeah, that's a good question. And so the ponds at Riverwalk, for those of you who aren't local, Riverwalk is a park that's right next to our most downstream site at the Stockdale Bridge. And there's some ponds there that water goes to, that's from the Kern River. And if I remember correctly, that's under the purview and management of the city of Bakersfield. And so there are fish in those ponds, and occasionally Cal Fish and Wildlife will do fish plantings there. I did check, and when we observed our rainbow trout the first time, it did not coincide with fish plantings of rainbow trout in the river, so that was kind of nice. And there is actually do have video. If you're interested, you can email me and I can share a video with you. But there is a grate. So as water goes into those ponds that river walk, that water then ends up and drains into the Kern River channel downstream of that pedestrian bridge where the Bellevue Weir is. And we do have some footage from a year ago of fish being stranded when the dewatering event happened. But since then, we have been walking most from Stockdale Bridge to that grate since the dewatering event, and I have not seen any fish from the ponds in the river since the dewatering event happened. And we go out once a month. Okay, it's not to mean that there aren't any, but I think it's just so little water that enters the river channel post dewatering that we just haven't seen any fish in that part of the river.